Yeah, so this is Trypanosomiasis gambiense, a protozoan parasite. Okay, it causes the fatal form of African sleeping sickness, uh, Gambian trypanosomiasis. Okay, so Trypanosoma gambiense is a protozoan parasite, and it is intercellular parasite. You remember? So generally, the antibody is considered as extracellular parasite. Okay, so trypanosoma is considered as intercellular parasite. So it lives in the human blood plasma between the cells and lymph. Okay, so lymph and the lymph tissue, clearly spleen. So it is confined to particularly the Trypanosoma gambiense is confined to particularly African countries, particularly Nigeria and Congo. So it is a digenetic parasite. That means it completes its life cycle in two hosts. The primary host is man. Okay, intermediate host or secondary host or vector is, this is very famous, CC fly. Its scientific name is Glossina palpalis. Okay, so you remember the name. CC fly is common name. Uh, Glossina palpalis is scientific name. Okay, so there is nothing much to explain. Just you remember the names, just like a revision. Okay, so the terminology you remember. Okay, wherever I stress, so that means that is important for example. Okay, its reservoir hosts are uh, generally there is an antelope called moo, is generally called as moo. So other reservoir hosts are pigs, buffaloes, reed bucks, which are restricted, which are African. Okay. Anyway, CC fly feeds upon blood of man as well as uh, you know it acts as a sorry, CC fly feeds upon blood and also the blood of reservoir host. Okay, so Trypanosoma gambiense was first discovered by a scientist called Ford in 1901. Sir Sir David Bruce discovered that sleeping sickness is transmitted by CC fly. Okay, so this is the parasite vector, and this is the microscopic image of the trypanosomas in blood. Okay, so three different forms of trypanosome. So CC fly, it uh, almost similar to you know housefly, but it bites human beings and other mammals and vertebrates. Okay. So anyway, this is the structure. Okay. So as we already discussed that, so this is unicellular organism, a single cell, and it is covered by a pellicle or a strong periplast. Okay, so this is considered as anterior, this is posterior. Okay, at the um, tip of the posterior region, there is a granule-like structure called kinetoplast, and this kinetoplast contain, an independent DNA. Okay, so and below this kinetoplast, there is a blepharoplast or basal granule, from which the flagellum arises. Flagellum, it spreads on its body, and it attains some what we call the it forms a membrane-like structure called undulating membrane. Okay, and after crossing its body, so it appears or it is free, just like a normal free flagell. Okay, and the nucleus is very clear. It is present in the middle of the body with an endosome or nucleolus. And valutin granules are a kind of granules present. Some amount of glycogen is also present as reserve food material. Okay, so this is simply, and there is a connection. Actually, it is not given in the diagram. The blepharoplast are basal granule, and the kinetoplast is connected by uh, you know a connection called rhizoplast. Okay, so that is the morphology. It is a microscopic animal, and generally these are the different kinds of forms of trypanosomes. So here this is Leishmania form, leptomonad form, tripedial form, trypanosome form. So trypanosoma is considered as polymorphic form. So in the blood, it appears in different shapes. So based on the relative shape to the particular, for example, Leishmania is a separate, protozoan parasite, but here it looks like Leishmania form, Leishmania species. So this form of trypanosoma is considered Leishmania form. Okay, and this is another form of the trypanosoma that looks like a leptomonas form of the Leishmania. So this is another second form, and this is trithedial form, and this is original trypanosoma form. So as it appears in different forms, so trypanosoma is considered as a polymorphic form, polymorphic parasite. Okay, yeah, that is I should, yeah. So kinetoplast contain an independent DNA. Right. So that is a cell granule rhizoblast. The nucleus, endosome. Yeah, contractile vacuole is not present in the cytoplasm. Valutin granules, a kind of food material is present, and nutrition is saprosaic. That means it absorbed food material from the flesh or the tissue of the host. So that is saprosaic here. Reproduction, only asexual reproduction is seen. That is particularly longitudinal binary fission. Okay, so you can see in the diagram uh, longitudinal binary fission. So a kind of asexual reproduction occurs. There is no sexual reproduction in trypanosoma gametes. Okay, so Yeah, as I have already told you, the life history or the trypanosoma is a digenetic parasite. It completes its life cycle in two hosts. The primary host is human being, secondary host is CC fly. Okay, so here you can see this is called metacyclic form. Okay, so the metacyclic form is present. See, this is metacyclic form that is present in the, you know, the salivary glands of the cecifla. When it bites a man, when it bites a man, okay, it enters, into the bloodstream of the bitten man. Okay, so this metacyclic form, it attains long and slender form. Okay, in the blood plasma, it undergoes many, many times longitudinal binary form. Okay, so later uh, it it uh, it transform into three forms actually. One is slender and long form, intermediate form, and short and stumpy form. Okay, there are three forms are formed from the newly formed trypanosoma. Okay, long and slender, intermediate, and short and stumpy form. So all these are, they are infective stage, or the next stage of the life cycle of the animal is continued only in CC fly or intermediate host. So when the infected man is bitten by another SCC fly. Okay, so all these forms, they reach the gut or midgut of the CC fly. So in the midgut, only this form, short and stumpy form, it survives. Okay, so the remaining forms are, they cannot withstand the pH and conditions of the midgut of the CC fly. So after some time, the short and stumpy form, again it attains long and slender form. Okay, it undergoes, it absorbs the food material from the midgut of the CC fly. Okay, and it undergoes asexual reproduction by, longitudinal binary fission. Okay, and increases in number. Okay, so then it attains another form called trithedial form. Okay, so here there is a small difference. The position of the this basal granule is approximately below the nucleus. Okay, and again this trithedial form, it divides, it pierces the, you know uh, midgut wall. Okay, and enters into bloodstream. Okay, so again it divides and is transformed into metacyclic form. So this metacyclic form is infective stage 2 man. Okay, so until this stage, it is in the midgut. Okay, so this actually slender and long form pierces the midgut wall. Okay, and reaches the, the coelomic cavity or bloodstream, and it reaches ultimately to the salivary glands, from where it enters into its main host, the human being. So this is life history uh, in brief. Okay, let's see. So first of all, CC fly, it is present in the salivary glands. So the form is metacyclic form. Okay, then CC fly bites man. It enters into human bloodstream. Okay, so it is the metacyclic form is converted into trypanosoma form in the human blood. Okay, and it undergoes longitudinal binary fission to form three kinds of form: long and slender form, short and stump form, and intermediate form. Okay, so the short and stump form is the infective stage to CC fly. Okay, and it can only survive in the CC flies midgut. Okay, so this form. Then, so such the infected man whenever bitten by CC fly. Okay, so it reaches to whenever it sucks the blood of the human being, the fly. So the different forms of the trypanosoma, they reach the midgut of the fly. Okay, short and stump forms survive. The remaining will be so they are diminished. Okay, so within 10 to 15 days, so the short and stump form it undergoes binary fission and produces slender and long form. Okay, so then from that it it, it pierces the midgut, okay, and reaches the glandular ventricle, okay, of the mm, fly, and from there it reaches to salivary gland. In the salivary gland, it again divides by binary fission, okay, to form criteria form. Okay, so the criteria form again divided by binary fission to form metacyclic form. So the metacyclic form is the infective stage to human being. So this metacyclic form we should remember. Okay, so which is infective stage to human being. So in this way, the life cycle again continues. Just follow the diagram, okay, and read once this sequence. So pathogenesis. So actually, the disease caused by the trypanosoma is generally, you can call it as trypanosomiasis. Okay, so all the symptoms and disease caused by the trypanosoma is simply called as trypanosomiasis. Okay, so whenever the infected fly bites, uh, there will be an itching at a bitten place. Okay, after reaching human blood, it causes a fever, a kind of fever called Gambia fever. Okay, so until it is in the blood, so the fever that occurs that happens in the man is called Gambia fever. Okay, so the symptoms of Gambia fever are anemia, headache, swelling of cervical lymph glands, and changes in spleen due to fever. Okay, so these are the symptoms. When it reaches cerebrospinal fluid, okay, so brain and spinal cord లో ఉండేటువంటి cerebrospinal fluid కి ఎప్పుడైతే the parasite reaches, okay, so that causes sleeping sickness. Okay, the person who suffers with the sleeping sickness, so he always tend to go to sleep for very, very longer time. Okay, so that is the main symptom of this, you know, uh, sleeping sickness or Gambia fever. Okay, so if at all not treated in time, there may be death occurs within sleep itself. Okay, so that is Gambia fever and sleeping. It is also, you can also call it as African sleeping sickness because it prevails mostly in African countries. Okay, so treatment for trypanosomiasis. These are the some of the drugs. Okay, like Bayer 205 pentamidine low medium drugs. So arsenic antimony drugs like arsamine, milacin oxide, etc. Uh, are the drugs to be used. So that is about the Trypanosoma gambians. And then next parasite is Plasmodium vivax, so which is a malarial parasite. So there is no particular, Plasmodium is a very, very, Plasmodium life history is very, very complex. There is no particular morphology, we cannot pinpoint this is Plasmodium, shape and structure. Okay, so there are different stages with the different structures and shapes. This is one of it. Okay, so in the RBC, the Plasmodium looks like this. Anyway, Plasmodium causes malaria fever. We all know that in man. So the term malaria was introduced by Marculoch, and malaria meaning. So in the name actually in earlier days the malaria fever was thought to be caused by bad air. So that's why the name Mala means bad, area means air. So they used to believe that the malaria was generally that is occurred by bad air, but that is not true. But the name was continued as it is. So Charles Laveran was the first scientist who discovered the parasite, this parasite in human blood in 1880. Later, Golgi also confirmed. He also noticed the presence of malarial parasites in human blood. Okay, so there is another scientist called Richard Pfeiffer. So he told that it is caused by biting of blood-sucking insects like mosquitoes. And then Sir Patrick Manson. Okay, so he told that cause of malaria is, so he precisely told that, so malaria, due to mosquito bite, the malaria is occurred. So this statement was given by Sir Patrick Manson. And the famous Sir Ronald Ross in 1897, so in Secunderabad. So when he was working in the British Army, he was a British researcher and doctor. So he discovered the oocysts, okay, in mosquito stomach wall. Okay, many oocysts he found. Okay, so later, so it was very clear that so from the oocyst, the malarial infective stage is produced, and that is infected to human being. So this is a very, very important, incident in regarding the malarial disease. So that's why he was awarded Nobel Prize in 1902. Okay. So Serrano Aldras, okay, that is in Hyderabad or Secunderabad. Right. So Grassi explained the history of Plasmodium in female mosquito. Okay. And then there is another scientist called Short. So he explained, you know, not completely, Short and Galgi also. Okay. So then, yeah. So what is Plasmodium? Plasmodium is a sporozoan intracellular parasite. So entamoeba is extracellular parasite, and Trypanosoma gambiense is considered as intercellular parasite, whereas Plasmodium is intracellular parasite and it belongs to the subphylum Sporozoa. Okay, so there are mainly there are four kinds of Plasmodium species: Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, Plasmodium falciparum. Okay, these are the there are other species are also there in the Plasmodium, but these are the famous malarial parasite species that causes different kinds of malaria, malarial fevers. Okay, so as usual, it is also a digenetic parasite. Here, slight change is the primary host is considered as female Anopheles mosquito. Remember, don't be confused. Why it is considered as primary host? Because even though it is its main life history occurs in human being, the sexual reproduction occurs in mosquito. That's why, so the mosquito is considered as primary host. And then secondary host is man, in which asexual reproduction only takes place. But in man, the number of the plasmodium increases in enormously. Okay, so its number is increased in very high number. So structure of the plasmodium. So this is the sporocyte of a you know um, plasmodium. Okay, so it is surrounded by an elastic membrane called uh, pellicle. Okay, so the cytoplasm is as usual. It contains all kind of cell organelles. Okay, so a clear uh, nucleus is present in the middle of the cell. Okay, and it contains the nucleolus also. Okay, so an interesting part is its shape is sickle shape. Okay, sporozoite shape is sickle shape అంటే కొడవలి ఆకారం అన్నమాట. So at the anterior region of the parasite, okay, so there is a structure called, this area is called, uh, apical cap. So from which one pair of organelles are hanged or present. Okay, so they're called rock rice, which are secretory organelles by which it penetrates the host cells. So that is the structure of the uh, malarial parasite, the infective stage, the sporocyte. Okay. So anyway, the sporocyte is the main stage of plasmodium. It is sickle shaped. Sickle shaped. Okay. The body is covered with pellicle. So anterior apical complex or apical cup. Apical cup or apical complex is present that can help for the penetration by its secretion. Okay, nucleus is present. So then life cycle in man. Right. So the sporocyte, which is an infective stage to human being that is present in the mosquito salivary glands as usual. Okay. So the sporocyte, it resides in the salivary glands of the mosquito. So when a mosquito with sporocyte bites a man, okay, so the sporocyte reach to human blood. Okay, so in the blood, according to Short and Granham, okay, so it wanders up to 30 minutes and then it reaches the liver. Okay, so due to the biting of the mosquito, female Anopheles mosquito, okay, so the infective stage, the sporocyte, it enters into human blood, it wanders up to 30 minutes. Okay, so and then it identifies the liver and it reaches the liver. It enters into liver through bloodstream. Okay, there are in human being, the life cycle three kinds of life cycles occur in man. Okay, so one is, I'll write here, pre-erythrocytic life cycle. Pre-erythrocytic life cycle, and the second one is exo-erythrocytic life cycle, and the third one is erythrocytic life cycle. So these are the three kinds of life cycles or cycles that occur in human being. Remember, in human being, only asexual reproduction takes place and the number of the parasites is increased. Okay, so first of all, let's see pre-erythrocytic cycle. See, this is the sporocyte. Okay, so due to biting uh, human being, it reaches the human blood and it reaches from the blood, it reaches to the hepatocyte, sporocyte entering a liver cell. Okay, that is hepatocyte. So in the hepatocyte, it eats the liver cell. Okay, the sporocyte increases in size, which is known as Shijat. Okay, so first of all, it is called trophocyte. Then, as it undergoes a multiple fission type of asexual reproduction, uh, this stage is called Shijat. Shijat produces almost 1,000, cryptozoites. Okay, so that is called pre-erythrocytic life history. So some of cryptozoites, okay, enters into, you know, another new, what we call the liver cell or hepatocyte. So these cryptozoites are generally, when they enter into a new liver cell, are generally called as metacryptozoites. So again, they eat this hepatocyte, cytoplasm and all those, okay, increase in size and undergoes schizogony, a multiple fission. Okay, and the lysis of the hepatocyte takes place, and they are released. So these are called, you know, cryptomerozytes actually. Okay, so cryptomerozytes, microcryptomerozytes, and uh, macrocryptomerozytes are formed. So here, uh, the micro, sorry, these are metacryptozytes. These are two types, micro and macro. Out of two types, the micro metacryptozytes, they enter RBC, or some of the cryptozytes may enter the RBC. Okay, so anyway, let's see the parts. Yeah, so pre-erythrocytic cycle. So it occurs in hepatocytes of liver. So in the liver, the parasite sporocyte undergoes asexual multiple fission called schizogony. So to form, so cryptozytes, they are called cryptozytes. So within up to 8 to 10 days, so these cryptozytes, they eat the hepatocyte, increase in size. Okay, so they attain a stage called cryptoshygot. Okay, so they undergo the schizogony to produce up to 1,000 cryptomerozytes. These are the cryptozytes. These are the cryptomerozytes. Okay, so at the end of the pre-erythrocytic cycle, approximately cryptomerozytes, about 1,000 cryptomerozytes are formed. So due to filling of the hepatocyte, the hepatocytes are so lysed or broken. Okay, so these cryptomerozytes, they either they enter into a new hepatocyte or RBC. So cryptomerozytes, you remember. So out of these cryptomerozytes, some are entered into new hepatocyte or some are reached to RBC. So the cryptomerozytes which enter a new hepatocyte, they follow, they perform exoerythrocytic uh, life cycle. It is also known as phanerogamy. Okay, in new hepatocyte, new hepatocyte rigid cryptomerozytes are called, so they are called metacryptomerozytes. Okay, so again they eat hepatocytes. Okay, to form metacryptoshygot, which undergo schizogony. Okay, to form 1,000 metacryptomerozytes. So there are two types of metacryptomerozytes: micrometacryptozytes and macrometacryptozytes. Micrometacryptozytes are small in size. Cryptomerozytes are, uh, you know, mm, larger than the micro metacryptomerozytes. Okay, in Plasmodium falciparum, the exoerythrocytic life cycle is not present. Only pre-erythrocytic life cycle is present. In remaining all three, the both pre-erythrocytic, exoerythrocytic life cycles are present. Anyway, so out of these two kinds of metacryptomerozytes, micrometacryptomerozytes, they start erythrocytic life cycle. You can also call it as cycle of Golgi. Okay, so macro metacryptomerozoites enter new hepatocytes. అవి పెద్దవి ఏవైతే ఉన్నాయో అవి కొత్త హెపటోసైట్స్ ని అవి అటాక్ చేస్తాయి. ఓకే, సో విత్ దీస్ టూ life cycle, okay, so the parasite increases its number. Okay, so what is the use of these two pre-erythrocytic and exoerythrocytic uh, life cycles? Okay, simply the parasite increases its number. Okay, now micrometacryptomerozoites are cryptozoites. Okay, so they enter, some of the cryptozoites enter the RBC. Okay, so here the third one is erythrocytic life cycle. It is also known as cycle of Golgi. Okay, so cryptomerozoites are micrometacryptomerozoites. Okay, so they start this life cycle. ఓకే, ఇందాక నేను చెప్పినటువంటి ఐదర్ ఈ క్రిప్టోజాయిడ్స్ కానీ లేదా క్రిప్టోమీరోజాయిడ్స్. క్రిప్టోజాయిడ్స్ ఆర్ మైక్రోమెటాక్రిప్టోమీరోజాయిడ్స్ ఏవైనా. ఓకే, దే స్టార్ట్ దిస్ ఎరిథ్రోసైటిక్ లైఫ్ సైకిల్. ఓకే, దే ఎంటర్ ఆర్బిసి. ఓకే, సో నౌ దే ఆర్ కాల్డ్ ట్రోఫోజాయిడ్స్. So this stage is called trophozoite stage. See, trophozoite. Okay, so then here, as it is increasing in its size, there is a big vacuole is formed within the parasite, and the nucleus, it reaches the periphery of the vacuole that looks like a ring. So this stage of the erythrocytic life cycle is called signet ring stage. Then this vacuole is disappeared. Okay, and the signet ring stage of the parasite, it attains pseudopodia, so that looks like an amoeba. Okay, so now this stage is called amoeboid stage. So now, with the help of pseudopodia, the parasite generally it engulfs the globin, hemoglobin. In the hemoglobin, the globin protein is consumed by the parasite, and heme is left by the parasite. So that heme becomes hematin, a kind of poisonous toxic substance. Hematin. Heme is normal, but hematin is a toxic substance. Okay, so anyway, this amoeboid stage, it undergoes erythrocytic schizogony. So that stage is called erythrocytic schizont. And in this stage, if you observe the RBCs, so many Schaffner granules are present. These Schaffner granules are, byproducts of heme. Okay, so later stage is generally called as rosette stage. So in which the here, due to division of the nucleus, okay, many small parasites are produced within the schizont. Okay, so they are called erythrocytic merozoites. What name are erythrocytic merozoites antam? These are actually, they arranged like a flower, like a rose flower. Okay, so within the RBCs, so that's why this stage should be the stage is rosette stage. So after rosette stage, so as the number is increased, the parasite number is increased in the RBC will be broken. Okay, so due to it, this all occurs within the blood vessel of the man. Okay, so the harmful hematin or hemozyme granules. Okay, so harmful hematin is nothing but hemozyme granule. So that is released into the plasma or blood of the man. So now, due to the hematin, so the malarial fever here it occurs. So anyway, these are the erythrocytic merocytes. Okay, so these erythrocytic merocytes, so they again attack or infect another new RBC, and the same, life cycle is continued. So in this way, this is another kind of schizophrenia or multiple fission. So to increase its number in the host. Okay, so that is erythrocytic life cycle or cycle of Golgi. Okay, so these are the points. Yeah, hemozyme granules cause malarial fever, so which gives chills in infected man. Okay, so the merocytes are released, infect pneumonitis. Okay, so due to this, the anemia. So anemia means less blood. Okay, so anemia is occurred. Anyway, so what is incubation period and how many days in plasmodium? So incubation period is the time taken for occurrence of malarial fever from parasites first entry into body. అంటే పారాసైట్ ఎప్పుడైతే దోమ కుట్టడం వల్ల బాడీలోకి ఎంటర్ అవుతుందో అప్పటి నుంచి మొదలుకొని మొదటిసారి ఫీవర్ వచ్చేంత వరకు ఉన్న గ్యాప్ పీరియడ్ ని మనము ఇంక్యుబేషన్ పీరియడ్ అంటాం. ఈ లోపు అది దాని నెంబర్ చాలా మొత్తంలో పెంచుకుంటుంది. సో ఇట్ టేక్స్ అబౌట్ 8 to 10 days in human being. Okay, so erythrocytic schizophrenia occurs many times in RBCs. Okay, in many RBCs many times. Okay, so the in, due to erythrocytic schizophrenia, many erythrocytic merocytes are formed. So out of which some of the merocytes they are transformed into gametocytes or gametes. సో ఇక్కడ మనకు ఫామ్ అయినటువంటి మెనీ merocytes or erythrocytic merocytes, they enter into a new RBC, that's common. But some of the merocytes, they are transformed into, they are transformed into, gametocytes. Okay, so this is smaller one, this is bigger one. Smaller one is called microgametocyte or male gametocyte. Okay, bigger one is, mega gametocyte or macro gametocyte. Okay, so in micro mega gametocytes, the nucleus size is larger when compared with the cell size. But in mega or macro gametocyte, so the nucleus is peripheral and smaller in size. And these, stages, micro and macro gametocytes. Okay, they are actually infective stages to, primary host, the mosquito ki avi infective stages and the gametocytes. Okay, not only gametocytes. Okay, so now this is in the blood of man it occurs. Okay, so here whenever a mosquito sucks the blood of the human being, these gametocytes, okay, with all these, different kinds of cryptocytes and cryptomerozytes and erythrocytic merozytes. Okay, and also these gametocytes. All those they enter into the stomach of the you know the mosquito. Okay, so now from the gametocytes, the gametes are formed. From the gametocytes, the gametes are formed. Okay, so the mega or macro gametocyte, it forms, you know, uh, mega gamete or female gamete. Okay, and the micro gametocyte, okay, overall it produces the micro gamete or male gametes. Okay, and here it is the fertilization is taking place. After fertilization, so the zygote is formed. So the earlier form is called ookinete, okay, which pierces the you know gut or stomach wall of the mosquito. Okay, so here this ookinete, it synthesizes, it prepares a thin sac around itself on the wall of the outside surface of the wall of the mosquito. Okay, now as it contain a cyst. Okay, so this stage is uh, famously known as oocyst. The oocyst, okay, again here asexual, first of all, after fertilization, okay, when it forms the oocyst, the first division it occurs is meiosis. So due to which only nucleus is divided, okay, and that all the nuclei that are formed here, okay, so they are unis, haploid in condition. Remember. So here, after the meiosis, many mitotic nuclear divisions takes place. So to form many nuclei, so these nuclei, each nuclei attains some portion of the cytoplasm of the, you know, ookinete. Okay, so then they are formed like this. So the primitive sporozoites are being formed in the next stage. Okay, so after number is increased, okay, this cyst is broken. Okay, so the cyst is broken. So these are generally considered as sporozoites. Okay, so these sporozoites which are released into the you know yeah, so coelomic fluid or blood. Okay, so they travel in the bloodstream and they reach the salivary gland of you know yeah, salivary gland of what we call the mosquito. Whenever such a mosquito with these sporozoites in its what we call the yeah, so the sporozoites they reach the salivary glands. Okay, whenever it bites a normal man and an infected man, the sporozoites which are infective stage to human being, they reach the human blood. So this total cycle was explained by Sir Ronald Ross. So that's why this sexual reproduction or life history of the Anopheles in mosquito is generally called as so cycle of Ross. మొత్తాన్ని Ross cycle. cycle of loss అని పిలుస్తాం. ఓకే, సో దట్ ఈస్ incubation period is for male microgametocytes, big nucleus, female macrogametocytes. So here in mosquito stomach, the formation of gametes from gametocytes is called, you know, gametogamy. Okay, the formation of gametes from gametocytes is called gametogamy. So when a mosquito bites an infected man, so all the merozoites enter into mosquito gut. First mosquito gut lo get. So all the forms are, you know, diminished in the gut of the mosquito. Only gametocytes are you know, survive. So in the stomach, these gametocytes, macro and micro. So the microgametocytes nucleus is divided into 2 to 3 times. So with that, the nucleus, each nucleus is divided into 2 to 3 times. So each free nucleus within the, you know, the gametocytes or microgametocyte, so attains some cytoplasm. That's why this is called schizophrenia. So, and here you can see, yeah. So this is how the male and female gametes are formed. These are the male gametes. This one is the female gametes. So here 2 to 3 times the nuclear division takes place. So, and each nucleus attains some amount of the, you know, plasma and cytoplasm also. Okay. So, and by a process called, it looks like each, you know, male gamete looks like a, what do you call it, flagellum. Okay. So now about 6 to 8 such kind of, you know, male gametes are formed. Okay, which are very active. Okay, they show, you know, metabolic movements or lashing movements. Okay, so anyway, here that is the formation of male male gametes. Okay, whereas in the ovum or macro cryptomerozoite, okay, so increases in size. Okay, so and the nucleus of the this macro metacryptomerozoite. Okay, so here it increases in size. Okay, and the nucleus is divided into two by, you know, meiotic division. Okay, here not meiotic division. Here the meiotic division. Anyway, the nucleus is divided into two, divided into two. Okay, one is nucleus is diminished. Okay, so that is considered as polar body, and the remaining nucleus, okay, with cytoplasm is considered as female gamete. The female gamete contains, you know, a fertilization cone through which the male gamete enters into the female gamete. So by a movement called lashing movement, the male gametes, okay, so they reach these, you know, female gamete, and at the cone of reception or fertilization cone, so this male gamete, so it enters or it fertilizes the ovum to form a zygote, which is considered as ookinete. Okay, so this ookinete, it pierces the stomach wall, and outside the stomach wall, uh, so it forms a cyst. Okay, so now this cyst containing stage without any, you know, uh, stages. Okay, so special stages that is called oocyst. Okay, so here the nucleus is always okay. It divides into many smaller nuclei. Okay, each nuclei attains some amount of cytoplasm to form many sickle-shaped sporozoites. Okay, so after filling up these oocysts, filling up the oocyst with a number of sporozoite. Okay, so this yellowish area is nothing but the hemocoel of the mosquito. Okay, so whenever it is released, okay, into the bloodstream. Okay, so the sporozoite and other stages of the mosquito. Okay, the whenever such sporozoite after releasing, they reach the salivary glands of the the parasite. Okay, so when such mosquito with parasite bites a man, these infective stages, which are trophozoites, they reach into the blood and then to the liver. So this is how the whole sexual reproduction takes place. So kainite, okay, after fertilization, the zygote is becomes little bit slender. Okay, that stage is called eukaryote. So, and it actively moves. Okay, so that stage is eukaryote. So it pierces the stomach wall, settle outside layer of the stomach. So it prepares a thin sac around itself. Now it is called oocyst or sporant. So it feeds on stomach wall cells, increase in size. Okay, in each stomach. Okay, there will be about 50 to 500 such oocysts are seen in an infected mosquito. So anyway, when it is increased, so now that stage is considered as sporoblast. The next heading is sporogamy. The sporoblast first undergoes meiotic division. ఎందుకంటే ఫర్టిలైజేషన్ జరిగింది కాబట్టి సో మియాటిక్ డివిజన్ కి జరుగుతుంది. ఓకే, సో నౌ ఫ్రమ్ నౌ ఆన్ the sporozoites to be formed, they are haploid. So anyway, then yeah, so after meiosis, the newly formed cells they undergo so mitotic cell division. Okay, many times. Okay, so that is a kind of multiplication. So that occurs in nucleus. Each nucleus attains cytoplasm. So many sporozoites are formed. Chala sporozoites are formed. So this stage, so that stage of the animal is called sporocyst. Okay, so the sporocyst, when it is filled with sporozoite, okay, yeah. So after lysis of the sporocyst, so many sporozoites are released into the blood of mosquito or hemocoelomic cavity of the mosquito. So from the blood, the sporocyte reaches the salivary glands. So this whole process, when from after fertilization, when the zygote nears the, on the stomach wall and forms a sporocyst, okay, and ultimately, so many sporozoites are formed. So this whole process is called sporogamy because sporozoites are being formed. The sporozoites are infective stage to human being. So anyway, when such mosquito with sporozoite in its salivary glands, okay, so when such a mosquito bites a normal man, okay, the sporozoites they enter the man, enter man. Okay, so man, you can say here blood. Okay, so anyway, that is how the infective stage, the sporozoites are formed. So Plasmodium causes malaria fever. Okay, its symptoms are Plasmodium vivax causes a malaria called benign tertian malaria. So prior to malarial fever, so when it is after completion of the erythrocytic cycle in the man, the malaria fever is formed or occurred. So prior to fever, 2 to 3 days, headaches, muscle pain, lack of appetite, ఆకలి లేకపోవడం. Okay, so all these are occurred. So after that, for every 48 hours, okay, the fever is caused. Okay, so here the temperature will reach up to 104 degree Fahrenheit. Okay, so anyway, the fever lasts for 6 to 10 hours. ఒకసారి ఫీవర్ వచ్చిందంటే 6 నుండి 10 గంటల వరకు ఫీవర్ అనేది కంటిన్యూ అవుతుంది. ఓకే, సో 10 hours తర్వాత 6 to 10 hours తర్వాత బాగా చెమటలు పోసి, ఓకే, సో స్వెట్టింగ్ వచ్చేసి ఫీవర్ ఈజ్ స్టాప్. మళ్ళీ 48 hours మళ్ళీ వస్తుంది. Okay, so it continues until we treat it. Okay, so due to the malarial parasites in the blood and liver of the human being, the person becomes weak. So treatment, these are the drugs like quinine, chloroquine, primapine, polydrine. Okay, all these are the some of the famous drugs that prevent, that cure the malaria fever. So precautions, what are the precautions we should take? Should keep our surroundings clean. We should use anti-mosquito sprays in dirty areas in our surroundings. So while sleeping, we should uh, apply odomous cream to our body or skin. So we should use mosquito nets and coils. Okay, so generally the mosquito reproduces in stagnant water. So in stagnant water, so larvivorous fishes are rare. So like Gambusia, Aphinis, okay, Lebestis fish. So these are the larvivorous fish. They eat insect larvae. Okay, so and also we should rear some special plants like Utricularia, which is generally larvivorous plant. Okay, So it is grown, within water, the Euclid plant. So anyway, so these are the precautions to be taken for the malarial fever, and these are the drugs for the treatment of the malaria. Right. So this is the table that shows, different kinds of properties of different species that cause malaria. Okay. Anyway, so here Plasmodium vivax causes benign malaria. Plasmodium ovale causes ovale tertian malaria, or you can also call it as mild tertian malaria. Okay, then Plasmodium malariae, quarter malaria. Okay, so and Plasmodium falciparum causes malignant malaria. So out of these four, species, Plasmodium falciparum is very, very dangerous. It may cause death to the infected males. And in Plasmodium vivax, it produces Schaffner dots in RBCs. Oval also Schaffner dots. Malaria produces Geman dots, different kinds of dots. And in falciparum, Marl's dots are present in the RBCs. So Plasmodium vivax, the time taken for the Shaijobani in the blood, okay, is 48 hours. 48 hours. In malaria, that is 72 hours. In falciparum, 36 to 48 hours. And spreading. Okay. So these are the some important, some points. Okay. Actually, incubation period also should be given. Okay, I will give it in the notes. Okay. So with this, the protozoan parasites part is completed. Okay, let's complete the protozoa phylum tomorrow.